All right, welcome back, Bio 153 students. And today we're going to continue talking about the immune system, focusing on the adaptive defenses. So this is all coming from chapter 22 in your textbook. So we're going to focus in on learning about antigens, which are really anything that's considered foreign in the body, along with learning about antibodies. So we're going to talk about the different classes of antibodies and how they help the immune system and how they go after and tag these foreign items, these antigens. We'll talk about vaccinations. And of course, we'll talk more about COVID-19 and the coronavirus as well. So a lot of cool things are coming up. And we'll dodge into them soon. And you can see even on this first slide here, this is a picture here of antibodies. These items here that are kind of V-shaped here, the yellow and blue structures here, and ultimately their goal is to tag antigens. In this case here, we have a virus right here, so they're going to tag it and marking it for destruction. So without further ado, here we go into our notes. Okay, so when we look at the adaptive defenses of the immune system, there are several important characteristics or functions that we should really key in on. The first one here, letter A, is the fact that this defense system is considered to be specific. It's considered to be targeted and ultimately directed towards particular pathogens. If that be a virus, if that would be a bacteria, if that would be a deadly fungi or a toxin, that ultimately is an important characteristic here. Letter B, another important function, the idea that this defense system is systemic. So, other words, referring to all throughout the body, not restricted to the initial infection site. And finally, letter C, the idea that there's actually memory. There's memory cells that can be built up after the initial exposure. So the initial exposure, if you can survive that and not die from it, you have ultimately built up of memory cells, a built up of antibodies now. And so important to know here too, that when we look at the adaptive defense system, it's considered to be your third line of defense. Your third line of defense. Okay, if we were to go ahead and look at the adaptive defense system, it is broken down into two major categories. So the first category, we are looking at what is known as humoral immunity. Humoral immunity. This is also known as antibody-mediated immunity, humoral immunity. This type of immunity really focuses in on the functions of the B cells or B lymphocytes. So a big emphasis on antibodies. And the function of these antibodies is that they can bind to foreign items, these extracellular pathogens like bacteria, fungi, parasites, bacterial toxins, viruses. And what they do is they're able to inactivate them so they don't do any more damage, and ultimately can mark them for destruction. And so they don't kill them, they inactivate them again, so they can't do any more harm in your body, and they put a marking on them. Just like if a horse or a cow was branded, if someone was out on a ranch. So they have this mark, this brand, and then ultimately phagocytes might see this. So like per se a macrophage, and then they can actually undergo phagocytosis and destroy the virus or the bacteria. Or likewise, complement also can go after per se bacteria and kill the bacteria. So something to consider there. All right, the next type of immunity here, the second category, is the cellular cell-mediated immunity. And the focus here is on T cells. So T lymphocytes. And they're important. Ultimately, they can kill foreign cells either directly or indirectly by enhancing the inflammatory response. And here we're looking at actual intracellular pathogens. So some of the targets here we're talking about would be cancer cells. They could go after maybe virus-infected or parasite-infected tissue cells, even cells of foreign grafts as well. So per se, a foreign graft would be maybe someone's a burn victim, the transferring of skin from another part of the body, and hopefully the body still accepts that, okay? Or we could even be referring to organs. Hopefully the organ is accepted and not considered to be a pathogen, an antigen. So that's the idea there. All right, so the big picture here, again, is the fact there's two types of immunity. We have the humoral immunity, which focuses on antibodies, B cells, and then cellular immunity, which is referring to T cells or the lymphocytes, T lymphocytes. Okay, so important to know those two major categories. All right, so this picture here, we are focusing in on the terminology here, looking at what's known as an antigen, a very important term to know, antigen. So the antigen is considered to be a living, or it could be a non-living type of substance that is recognized as foreign by the immune system and actually will activate the immune system. And this antigen, again, like I said, living or non-living, it can be natural, it can be synthetic, and even based off of the molecular structure of the antigen, it can be either complete or incomplete. So there's a lot of different things then that can be an antigen. Some of these might be no-brainers, like bacteria or toxins, different microbes, viruses, but other things too, cancer cells. Someone gets the wrong blood type when they're getting a blood transfusion. Even different plants where you have an overall allergic attack, like poison ivy or poison sumac or poison oak would be an antigen because of the response of inflammation. It's a foreign item in the body. And it can be other things too. It could be simply metals. Some people are allergic to different types of metals like nickel, and depending upon the jewelry that you wear, that would be an antigen because of the breakdown that occurs in your body. So there's a lot of different things that can ultimately be considered an antigen. Now, around the antigen, like we're seeing right here in this picture, here's our antigen, right? We have these antigen binding sites that we're seeing here, and ultimately right here would be an antigen binding site. This is an antigen binding site, etc., antigen binding site. And it's that binding site ultimately where the antibody can bind to. So again, this V-shaped structure is your antibody. Here we have an antibody. So this is kind of a docking zone for the antibody with the antigen itself. Another important term to look at is what's known as the antigen antigenic determinants. So antigenic determinants, ultimately here, we are looking at the location where free antibodies or even a lymphocyte receptor bind to the antigen. And you can see there's a variety of different types of shapes here. And so certain antibodies can only bind to that particular location. Very similar to the concept of having many keys on your keychain, and there's only one key that opens the door to your house. There's only one key that's designed to open up your car. Same type of idea. There's particular antibody classes, and only certain antibodies can actually bind on to a particular antigen. This is kind of a very simplistic view here, and we get the idea here of the antigen, like the bacterium that we're seeing right here in the orange. And then here we have the antibody right here. And this antibody is designed to dock and bind on to this bacterium. Just like over here, example, virus. Here we have a virus here. And this particular antibody is designed to only have an interaction with this virus to go ahead and tag it for destruction. Okay, so we have our antigen, like the bacterium or the virus. Okay, that would be your antigen here. So antigen versus down here, these structures that are the V-shaped would be an example of an antibody. Each one of these is an antibody. If we look at both of them, antibodies. Okay, so here we have the antibody. Okay, so we have the antibody here versus the antigen shown at the top. So you can see that here. This is a little messy. We can fix this here for you. So again, down here, we're looking at our antibody here. So again, antibody and antibody. Okay, so we're now going to take a look at different types of antigens, and really there's two major categories that we're going to focus in on. First, we're going to learn about complete antigens. So a complete antigen, typically we're looking at items such as different pathogens, bacteria, fungi, viruses, but even items such as pollen grains. And they have important functions. They have the property of immunogenicity, so the ability to stimulate proliferation of particular lymphocytes and antibodies, and the property of reactivity, so they can have the ability to react with products of activated lymphocytes, and hence antibodies released. The way to identify that you have a complete antigen ultimately is you have to look at the molecular structure. So here we have a complete antigen. This is the structure here. Based off this molecular structure of the complete antigen, it has the ability to dock right on to the antibody. So again, this is the antibody I'm circling here. And based off the structure, it has at least two antigenetic determinant sites. Here's a lovely picture here of the death cap mushroom. A great example of a complete antigen there. This mushroom, the common name is death cap because of the toxicity found in this mushroom. Thankfully, it doesn't grow in Michigan. It grows out of the Pacific Northwest, these mushrooms here, out in Oregon, Washington State, British Columbia, Northern California. And if you were to do a poor job of IDing your mushrooms and eat this, you would do a lot of damage to your hepatocytes, to your liver. So if a cat ate this by accident or a dog, they'd probably die. So a lot of toxins in that. We now take a look at another major category of antigens, And here we're taking a look at what are known as incomplete antigens. Incomplete antigens. And these are also known as haptens, just another name. So ultimately, this is really focused in on different types of drugs, or different types of medications, steroids. And so here are some of our examples. So penicillin, so a great example there for medication. Poison ivy, we've mentioned that. Animal dander, that might come from a dog or a cat. Different detergent products, cosmetic products, common household products, etc. And sometimes these can lead to a very harmful attack. And so when we look at the molecular structure of an incomplete antigen shown down below, here we have the incomplete antigen, again, another name, just hapten, and it's not able to initially bind right onto the antibody. So there's typically a carrier molecule that binds on, and together then, now they can go ahead and have this interaction with the antibody shown in the purple Y there. So again, great example of the incomplete antigen, poison ivy. This is a picture here of poison ivy. So this is very lovely. The 3-leaf plant found in the woods. So some people are very allergic to the poison ivy. It's the oil, really. If you realize you've touched the poison ivy and got oil on, it's very important to wash down that area of your body as soon as possible. If not, over time, the oil spread. You can break out into hives, which is extremely itchy, and you're probably going to have to be put on a steroid for sure to eventually try to take care of the poison ivy, like protozoan. So, okay. We shift gears. We've looked at two major categories of antigens, but now we're going to look at two major types of lymphocytes that are important to know. So first, we're going to take a look at B lymphocytes, also known as B cells. Again, they play a very important role with humoral immunity. The origin of where we find the B cells is the red bone marrow. So that's where they're produced, is the red bone marrow of the body, the B cells, the B lymphocytes. And then when it comes to the location of maturation, that would be the red bone marrow, where they mature and essentially are going to be maybe more classified as an adult. They've been educated to work in the immune system. The other classification for lymphocytes, we have what are known as T lymphocytes, or T cells. And they play a very important role with cell-mediated immunity. The origin of the T cell is going to be the red bone marrow. So the location of production, the same as B cells. But the location of maturation is different. It's going to be the thymus gland. Okay, the thymus gland. So a very important lymphoid organ that we, again, briefly mentioned when we looked over the lymphatic system. We're now going to take a look at the lymphocytes' development, their educational process. And this is important to note because we get an idea of different traits they develop, and then they're pretty much activated to work in your body. And T cells and B cells are critical for the immune system response and overall health and well-being. So the first trait here to be familiar with, number one, is the idea of immunocompetence. Immunocompetence. And this is the idea that these lymphocytes can recognize and bind to a particular type of antigen. Okay? They're able to recognize, and they can bind to a virus, or they can bind to a bacterium or a parasite or whatever it might be. So an immunocompetence is a very important trait. The idea that these lymphocytes become competent. They can do their particular job in your body. Number two, another important trait to be familiar with is the idea of self-tolerance. Self-tolerance. This is very critical. Very important for these lymphocytes to be unresponsive to self-antigens. In other words, very critical that these lymphocytes don't attack your body. Because when we're talking about lymphocytes that don't follow this property of self-tolerance and they attack your own cells, we're ultimately looking at autoimmune diseases. Okay? We're looking at fibromyalgia. We're looking at lupus. Okay? We're looking at MS and etc. So very, very important property. Both of these traits critical. And so these lymphocytes, B cells and T cells, are naive. And they're considered naive early on before they've ever had an encounter with an antigen. So they're considered to be unexposed to antigens. They haven't had that encounter yet. They're naive to the human body. They're naive to these foreign invaders. But eventually they'll have their encounter. Typically, they'll travel to secondary lymphoid organs. So they may go ahead and leave the thymus gland in case of the T cells. The B cells will leave the red bone marrow, and they go to secondary lymphoid organs. So they might go to the spleen, maybe they'll go to lymph nodes, maybe they'll go to the tonsils, etc. And then they may eventually have that first encounter with an antigen, and then they understand their role in your immune system. Okay, here's another picture looking at overall lymphocyte development. So again, very important, the idea of the origin of these lymphocytes. So the origin here, again, is going to be the red bone marrow. So again, that's going to be the case for the B cells. And likewise also the T cells. So the importance of the origin of where they're produced. But then you'll see they go to different locations when it comes to the maturation process. So the thymus gland, that's where the T cells are going to undergo maturation. Okay, that's where the T cells travel to. The B cells, however, aren't going to be traveling a whole lot. They're staying in the red bone marrow. Maybe they'll go to one other side of the bone or whatever, but they're going to stay in the red bone marrow when it comes to these B cells. Okay, so they stay in the red bone marrow. So that's where they undergo their maturation. This is where they'll develop, again, the property of immune competence. They'll become competent, okay? It's like they go through this educational process to be competent so that they're able to go ahead and properly encounter the antigen, identify it, know that it's foreign. And so that's very important, again, this idea of immunocompetence. And then the idea, again, of self-tolerance. So that's very important too. Okay, so immunocompetence, self-tolerance, again, so they don't attack your own cells of the body. So again, that's not good at all, because then we're talking about, again, autoimmune diseases. Again, like I've mentioned before, with lupus and fibromyalgia and MS and etc. So they develop this, and now they're still considered to be naive cells. But eventually, as they leave, B cells leaving the red bone marrow, T cells leaving the thymus gland, and now they're moving through your blood. They're moving through your lymph and your lymphatic system. And eventually, they may have that first encounter with an antigen. And so when this happens, the antigen encounter and lymphocytes, antigen receptors. So the receptors here will bind to the antigen. Here's the antigen here, and the red here looks like maybe a little snowflake, and in the middle is that lymphocyte. And based off of that, lymphocytes become activated. And they become activated. They've been identified to the antigen. They've detected that this is a foreign item in the body. And based off of activation, this can lead to a significant increase in the lymphocytes. So you have many that develop. You have all these clones that develop. It's almost like a clone war, like in Star Wars. So you have all these lymphocytes being produced, these clones that start to be developed. And eventually, in time, memory cells can be developed and effector cells can be developed. And hence, in time, at least with B cells, antibody secretion can occur. So this gives you a little bit of an idea of how lymphocyte development takes place. We're going to spend a good amount of time today focusing on humoral immunity. That's focusing on the work of our B lymphocytes, or B cells. So looking at humoral immunity, and let's say you're exposed to a virus. So let's say the virus that you're exposed to here is chickenpox, the chickenpox virus. Of course, there's a vaccination for that now. But back in the day when I was a kid, there was no vaccination to chickenpox back in the 1980s. I ended up getting the virus when I was in first grade, or was it second grade? Second grade, back in 1987. And my friend coughed on me, she had it, and then I got it. And with the chickenpox virus, the big thing is the fact that you end up with these red spots all over your body. They're very itchy, and it's just an annoyance. And literally took me about 2 weeks to get over it eventually. Thankfully, I didn't have too much homework to make up in the second grade. And pretty much, you get it. Can't go back to school. You have to quarantine for 2 weeks. And of course, back in the day, there used to be chickenpox parties. You know, bring your friends over. Everyone gets the chickenpox. They can quarantine together, you know. But it can be very severe. I had it pretty bad, and some cases led to death. But not a fun thing. Thankfully, now there's a vaccine for this. But when we look at the response to a virus, there's two major types of responses. There's a primary response and a secondary response. So what we're looking at here first, if you had the interaction with the chickenpox virus, we're looking at the primary response here first. Because the fact that there's no memory cells, no recollection to this virus, the overall response time of your immune system to this foreign item is slow. So we look at the step-by-step process here. So here we have the antigen. Again, this would be the chickenpox virus as an example. And eventually you might have some B lymphocytes nearby, and the antigen then can bind to the receptor site. So the lymphocyte comes, binds to the receptor site. The lymphocyte, this guy right here, is able to detect the antigen, and eventually you have activated B cells, and you form some clones right here, clones. Of the clones, some might become a memory cell. So this is representative of a memory B cell. Others will develop into what are known as plasma cells. So these are considered effector B cells, the plasma cells. And these plasma cells, they ultimately produce the antibodies, okay? So these are the antibodies right here. But the process of developing the antibodies is a very, very slow process. And ultimately then, these antibodies can move through your blood, lymph in your lymphatic system, and they can bind to free antigens. They can mark them for destruction. And then again, what will destroy the virus would be things such as phagocytes going after the virus. Definitely interferons can come in and isolate the damage of the virus. Natural killer cells can come in and kill them, etc. So this is the primary response. The big thing to get out of this picture is that the response time is very, very slow here. So here's a picture of the chickenpox virus. So lots of fun. You can imagine me having this when I was in the second grade, and my mom would always tell me, Chris, don't itch those scars, those red dots here, because it's very itchy, and if you start going ahead and they're popping up all over your body here, and you're getting them all over, eventually you may not have a happy smiley face anymore, because it might be very, very irritating, and you might ultimately have more of a frown because you have the virus. And of course, you itch them, you end up with scars. And so, again, you get them all over your body. So imagine some of these lovely welts. You get them all over your body, inside your mouth, in many locations. Some people get it worse than others. And once you have the virus in you, it's in your blood, it's in your body forever. And sometimes it can be reactivated later in life, and it can develop into a resurgence and develop into what's known as shingles. So maybe you've heard of shingles before. Typically, this can be reactivated, the chickenpox virus, into shingles, which is very similar. Typically, when you're older, elderly, you can develop the shingles. Stress can really bring on shingles. Sometimes some women can end up with shingles when they're pregnant because of the stress of the pregnancy, and they can develop shingles. So stress can really bring that on. And again, people that get shingles, it's the result that they have the chickenpox virus in their body. So, because once you get a virus, it's in it. Pretty much, it's that's in your body forever. Coronavirus. How can we not talk about corona? So the coronavirus, again, we hear about this all the time in the news. Right? It came over from China, and looks like it was first in our country in January, early January, and then eventually it went rapid throughout the country. It started over in California, the first case, and then Washington State, and then made its way eventually throughout all the states, and to Alaska, Hawaii, U.S. territories, and obviously in Michigan. Of course, in March it got really bad in March of 2020, and of course then for several months many things were shut down in order to limit the spread of COVID-19. You do what you can to reduce the chance of getting the coronavirus. Of course, wearing the mask. That's very key, to wear the mask, like we see here. That reduces the chance of COVID significantly, is to wear that mask. Because, like many viruses, it's airborne. So if you don't wear the mask, and you're walking, and you're within, less than 6 feet, and you're, laughing, you're talking a lot, and coughing, and all those things, sneezing, coronavirus can easily go out of your body and go into someone else. It's very, very contagious, COVID. So wearing the mask is important. Establishing social distancing. Washing your hands as much as possible, because if you don't and you're touching surfaces and things, someone who has COVID, and then you put your fingers into your eyes or your mouth or whatever, you probably end up with a coronavirus more likely than not. So it's very important to do what you can to be safe. Very key to practice the social distancing. Wear the mask at all times. Very, very important when you're around others. Eventually, hopefully, a vaccine will be out there. And hopefully that will come sooner rather than later. But you want to be safe. Many people have died. So hundreds and thousands have died. So it's very important to be safe. Even young kids have died from the coronavirus. All ages have died from the coronavirus. But of course, the elderly are more susceptible to coronavirus, because people with pre-existing conditions are more likely to have it the worst, because they're obese, or they have diabetes, or they have cancer, high blood pressure, or whatever it might be, and they're more susceptible to having very severe symptoms of coronavirus. Of course, some people are completely asymptomatic, and there's even research out there that even maybe your blood type, certain blood types might be susceptible to showing a lot more full extent of the coronavirus versus other blood types. So a lot of unknowns with the coronavirus, and you definitely want to be careful and be safe, because there's no vaccination for the coronavirus right now. but hopefully soon there will be. Here's a video, so if you'd like to watch this at home, this shows you a great 3D perspective of how coronavirus, COVID-19, is spread from one person to another, and how it eventually, the virus can go through your body. And again, many of the different symptoms. So, symptoms of the coronavirus, fever, fatigue, loss of taste and smell, severe inflammation of organs, pneumonia in the lungs. The biggest thing is ultimately the inflammation of the lungs and pneumonia, which eventually leads to death, is the shutting down of the lungs. And there's always, some people will make a recovery, but their lungs may not ever be the same. And there's even data out there showing that it could do harm to your other organs as well. Like even your heart. Your heart may not ever be the same again. It might lead to damage of your heart muscle. So very interesting. Goes through vaccinations, testing, etc. So it's a very interesting video, and I highly recommend for you to take a look at that. I think you might enjoy it. All right. So looking at the secondary response now. We've looked at the primary response. So that's everyone who's getting coronavirus right now. It's the primary response because, again, no vaccine, right? Some people might have antibodies built up. Some people do, some people don't. All based off the antibody testing that's available for some people. Okay, of course, you can tell if you have it, a positive reading based off of a swab test. But here we look at the secondary response now. So with the secondary response, this isn't nearly as brutal as the primary response if you are introduced to a virus. So the response time to plasma cells being formed and antibodies produced is much faster, 2,000 molecules per second for 4 to 5 days. So significantly faster than the primary response, which literally could take a week, week and a half or more. So we can see that here for that secondary response, you would also get this if you were vaccinated to like chickenpox and were exposed to somebody like that. You wouldn't have it nearly as severe. So we can see all these plasma cells being formed here and all these antibody molecules being produced here. And ultimately, these memory B cells provide that overall immunological memory and a fast response to that same antigen, like chickenpox, for example, or even polio. And a lot of that is, again, the vaccination, where you just have the secondary response, or the fact that years later, maybe you never got vaccinated, but years later you are exposed to the same virus again, and it's not as bad. Okay, this graph here is looking at the differences between the primary and secondary humoral responses. So the first part of the graph here, we're looking at the primary response. Okay, we're looking at the primary response right here to an antigen, maybe like the chickenpox virus. And we can see here that it's a slow process here. The lag period is 3 to 6 days. Peak levels of plasma antibody production, 10 days, week and a half. So we can see that on the scale here, pretty slow process. So this is showing the days, the time and days of antibodies being produced to go after the virus. For your x-axis down here, your y-axis is looking at ultimately the amount of the antibody production. So primary response very slow here. But what we see here then over here is the secondary response time. The case with someone who has the vaccination or they're exposed to the virus again years later. And now what we see as the antibody levels now peak in 2 to 3 days to play a huge role in going after the virus in comparison to the primary response, which took a week and a half. So, and these levels can remain high for weeks to even months. and memory cells responding within hours. So significant differences between the primary immune response versus the secondary immune response. Okay, we're now going to take a look at vaccinations. Vaccinations are so critical. I can't stress it enough. They play a huge role in ultimately confining a deadly virus, confining a pandemic, isolating it, eradicating the virus from a country. There are many vaccinations that maybe you had when you were a child. Maybe you had the polio vaccination, or smallpox, or measles, or others. Maybe you have the chickenpox one. And ultimately, the vaccination can spare us from the symptoms of the primary response, which can be very deadly with many of these viruses. Take, for example, this child down here who has polio. The polio virus is very devastating to the body. And you still have polio in many third world countries, in Africa, for an example, or in third world countries in Southeast Asia, for example. And what the polio virus can do ultimately is just totally destroy your muscles, and can leave you paralyzed, and it can weaken your heart muscle and all those types of things. So here she is paralyzed, cannot walk because of the polio virus. Before the vaccine was developed, polio was rampant throughout the United States. And many people contracted it and died. My grandfather had polio when he was a child in the 1920s. He was bedridden for a whole year. Thankfully, he wasn't paralyzed, but it did weaken his heart. And it did lead to one of his legs being longer than the other. And he didn't live very long because of polio, and actually ended up dying from a massive heart attack because of a weakened heart most of his life. Even President FDR had polio, and his whole presidency he was in a wheelchair because he could only somewhat walk when it came to his braces that he had on. And he contracted the polio virus when he was in his mid-30s, in New York State. So very deadly, these viruses, and very important to have vaccinations. Going into the medical field, it's very important to educate patients, educating people in the community about the importance of vaccinations. And hopefully one day we'll have a vaccination for COVID-19. We may not have one now, but hopefully soon a vaccination will be out there, one that is considered safe, lots of clinical trials, and can be approved for everyone to get. And important that everyone gets that vaccination for the coronavirus. We're now going to take a look at some differences of humoral immunity. We can break humoral immunity into different categories. So we can break this down into active versus passive humoral immunity. So, when we first look at the active humoral immunity, this is where there's actually overall immunological memory. So, active, actual memory. And this can be either naturally acquired or artificially acquired. So active, naturally acquired would be simply you get an infection, you've been in contact with a bacteria, the virus, you've been in contact with the coronavirus, COVID-19, and eventually if you survive the primary response of COVID, you now will have memory cells built up, antibodies. And so if you get a second encounter, it won't be nearly as bad. So that's the idea of active naturally acquired. Active artificially acquired is vaccination. You go ahead, you have the vaccination for the virus. You get the vaccination for the coronavirus. You get the vaccination for polio, measles, etc., smallpox. And memory is involved here for more long term. The difference over here with humoral immunity passive, the fact that the passive humoral immunity, overall immunological memory does not occur, and protection is a very short-lived event. Naturally acquired example would be the mother breastfeeding the child, where you have some antibodies passed from the mom ultimately during breastfeeding, or passed from mother to fetus in the placenta. Artificially acquired would be an example more of an injection of an immune serum. The case being perhaps for someone that is out hiking out west and there's a rattlesnake on the hiking trail and you sat down to take a break, maybe a water break, and you didn't see the snake and it came and it bit you. And now you have that venom in you. You could die and you need to take that anti-venom to go ahead and survive. So that would be an example of more of a passive artificially acquired. So definitely important to know this information. We're now going to take a look at antibodies. And this picture here, we're looking at a basic antibody structure. So when we look at an antibody, you always have at the top this antigen binding site. This is where the actual binding will occur with the antigen, with the virus, with the bacterium, whatever it might be. So there and over here would be those binding locations. And ultimately, we have the shape being Y-shaped, but it could be T-shaped as well, made up of polypeptide chains. And you have a heavy chain and you have a light chain, and structurally sound because of these yellow structures that you see here, these disulfide bonds. There's a hinge region and a stem region. Now the stem region is pretty important because the stem region ultimately here of the constant region will determine the antibody class. And there's actually several different antibody classes that all have different functions in your body. There are actually 5 classes, 5 immunoglobulin classes. So a fancier term for antibodies is immunoglobulins, Igs. So ultimately secreted by plasma cells. So that's just a fancy term when we start looking at antibodies, is referring to them as immunoglobulins, Ig. So we'll take a look at that next. Again, this just gives you a brief idea of the structure of the antibody. Okay, we're now going to run through the antibody classes. And it's very important for you to know these, as you might see them maybe on the next exam or a quiz. So very important that you know these. Know the functions. The first one here we have is IgM. Structurally a fairly big antibody, known as a pentamer. And here we can see that right here. You can see some of the important functions. It's typically the first antibody released and plays a big role with fixing and activating complement. And that's, of course, very important when you think of the activation of that blood protein complement with the classical pathway. Complement, very important for going and killing bacteria. Another very important class to know is IgA. So this antibody we usually find in mucus and other secretions in our body. So it's in sweat, it's in saliva, it's in milk, it can be in intestinal juice, okay? And it can ultimately prevent entry of pathogens, maybe trying to penetrate through the body. So again, bacteria, fungi, things along those lines. So we have IgM and IgA. Other antibody classes that you need to know. At the top, we have IgD. So IgD, the function is a little unclear. Based off of some studies that I have read, it might be able to react with specific bacterial proteins. But ultimately, we're not very clear on what IgD does. We're not entirely clear. And again, this is ultimately the structure there. The next antibody class we see here is IgG. So this would be 75 to 85% of antibodies in plasma. This is considered to be the main antibody of secondary and late primary responses. And it can also readily fix and activate complement as well. So again, in this picture, we can see that antibody class right there. And then the last one of antibody classes for you to know is IgE. This is very important, this antibody, when someone has a severe allergic attack. So maybe they have an attack from bees or hornets or wasps, or maybe they're allergic to food, maybe they're allergic to peanuts or seafood, okay? And also plays a role with chronic parasitic infections of the GI tract. Ultimately, this causes basophils and mast cells to release histamine, which again takes place during the allergic response for severe inflammation. So another very important antibody to be aware of, and there's IgE right there. Okay, so make sure to know your antibody classes. That's a very important item that you need to know. We now take a look at the overall mechanisms of antibody action. So again, for review, well, what do antibodies do in the body? Ultimately, their function, antibodies, is to inactivate and tag antigens, these foreign items in the body, toxins and viruses and parasites and bacteria and so on and so forth. And so, again, the antigen can come together with the antibody. The antibody comes to the antigen and a complex is formed. And we're looking here at 4 types of defenses that normally can occur based off of this complex which is formed. One type of defense is what's known as neutralization. So with neutralization, this is important. This can go ahead and mask dangerous parts of bacterial exotoxins. It can play a big role with masking dangerous areas of the virus per se. So very key, and we can see this here. These antibodies here are blocking these sites on the viruses and bacterial exotoxins. So that's one type of defense here, is the neutralization. Another type of defense is what's known as agglutination when we think of blood. So this usually comes with the clumping of mismatched blood. So that's an overall defense. The third line of defense is known as precipitation. So this usually would take place with soluble types of antigens that may be in the body. And you end up with a cross-linkage of many antibodies cross-linked to one another with antigens. And then the last type of defense, probably one of the most important and main one, is complement. So the complement, this blood protein, is considered the main antibody defense because it can lead to cell lysis, okay, of ultimately going after and killing bacteria again. We mentioned that in part 1 of our notes here. So cell lysis, again, important for killing of bacteria. So killing of bacteria, right? And then likewise, the complement can also play a role with inflammation. It can play a role with phagocytosis because here the complement can be found on the outside surface of the macrophage, the phagocyte, or the neutrophil, and that coating can be sticky, and that can lead to the foreign item, the antigen, the virus, the bacterium being stuck there, and then ultimately overall breakdown of the foreign item. And not to forget, the agglutination, again, can enhance phagocytosis as well. So remember your 4 types of defenses again: neutralization, agglutination, precipitation, and complement. We move on from humoral immunity, and now the next couple slides is going to focus on understanding of cell-mediated immunity. So cell-mediated immunity, or the cell-mediated immune response, ultimately focuses on the work of T cells or T lymphocytes. So we're going to learn a little bit about this process here. It's a very complicated process, so we limit the time we look at this. There are a diversity of different types of T cells, but major types we have what are known as CD4 cells and CD8 cells. The CD refers to cell differentiation glycoproteins, so probably sounds like a mouthful. The CD4 cells have the capability of evolving into what are known as helper T cells when activation occurs, while the CD8 cells can evolve into what are known as cytotoxic T cells, and they can go ahead and destroy cells harboring foreign antigens. There are other types of T cells, such as regulatory T cells, and there are also, of course, memory T cells, just like we had memory B cells. So again, they have that recollection of the antigen encounter. Some more terminology to be familiar with. We look at these very unique proteins, which are called the MHC proteins. So the abbreviation MHC stands for the Major Histocompatibility Complex. Major Histocompatibility Complex. And you're probably thinking, whoa, that sounds like a mouthful for sure. So there are 2 classes of these proteins. There's the class 1 MHC proteins and the class 2 MHC proteins. The class 1 MHC proteins are ultimately found on virtually all body cells, the exception being red blood cells, your erythrocytes. The class 2 MHC proteins are found only on certain cells in the immune response, such as B cells or dendritic cells. And then, when it comes to ultimately identification of the antigen, when we look at cell-mediated immunity, it's also important to be aware of APCs, antigen-presenting cells. So as the term refers to, these cells ultimately are presenting the antigen to another cell. So let's take a look and see how this process works. We're probably thinking, whoa, Chris, this is pretty crazy here. So let's take a look here. Here we have an antigen right here. So here we have a foreign item. So again, maybe we're looking at bacteria as an example. So maybe we look at that. And that's the antigen, right? And then what happens is the purple here represents your macrophage. So phagocytosis will occur. And ultimately, we go through the process of phagocytosis, and the bacteria is broken down into a small peptide fragment. So here's a small fragment. The class 2 MHC protein actually will have a receptor, will actually be found on the surface here of the macrophage. And there's a docking spot where the fragment of the bacterium would be, such as here. So it's found within the docking of the class 2 MHC protein. So that class 2 MHC protein here literally acts like a dock, and I guess there's one way to think about that. But then along comes the CD4 cell. So a CD4 cell, it's a T cell, right? And it has a receptor site. And this receptor can dock right where the class 2 MHC protein is that contains the peptide fragment of the bacteria. And based off of that, the CD4 cell understands that, oh, yes, I'm supposed to go and kill bacteria because it recognized that foreign antigen. I need to kill bacteria and destroy it in the human body. So it becomes activated, and then eventually more clones are formed, and you have all these clones which are going to go around in the body. And of course, they become helper T cells. This other example over here we have another antigen-presenting cell here. And maybe in this case, maybe the foreign item here, the antigen, is cancer cell. So we can just call this a cancer cell here. We'll erase this here. So let's say we have a cancer cell. So here we have a cancer cell. So maybe we're looking at cancer cells that are associated with leukemia or lymphoma or something along those lines. Again, this can be broken down, this cancer cell. Maybe it's phagocytosis that takes it out, okay? And you're left with a fragment, this peptide here, a fragment of the cancer cell. And it's able to dock in the docking spot because of the class 1 MHC protein. But then comes along your CD8 cell, which again can become a cytotoxic T cell, and it has T cell receptor site. And it's able to go ahead and dock on to where the peptide fragment is. And based off of that interaction, the CD8 cell knows now that, oh, I need to kill cancer cells. So I need to kill cancer cells. That's what I got to go after. So, based off the interaction then, you're going to have a huge army formed of clones. And they're programmed to go, they detect that cancer cell and destroy it. And that's the major target that cytotoxic T cells go after in the body, are cancer cells. Okay? All right, so the two major cells that are formed. Helper T cells we'll look at first. So helper T cells, again, they evolve from CD4 cells, and they play a role. They can help activate T and B cells. They can also induce T and B cell proliferation, so they can assist with an increase in the number of these lymphocytes. And they even can activate macrophages and recruit other immune cells, which can assist, of course, because they can undergo phagocytosis of a lot of these foreign invaders. These cytotoxic T cells, very important. They evolved from the CD8 cells, and they can directly attack and kill other cells if need be. And they can circulate in your blood. They can move through your lymphatic system and lymph fluid. And there's many targets that the cytotoxic T cells go after, but a major one they go after are cancer cells. There are others listed here that they can go after, so they can go after transplants. Maybe that's considered they identify that as foreign, so some people get transplants, okay, when it comes to the breast. So sometimes that's considered foreign. Could be virus-infected cells, okay? It could be cells within bacteria or even parasites too. But a major target, like I've mentioned, that they can go after ultimately here is cancer cells. And we see that even with the picture that I have here for you, right over here. Here's the cytotoxic T cell in the blue. And here they're directly going after to kill that cancer cell. Cytotoxic T cells play a big role with going after cancer cells in the body. And that's why when it comes to many different forms of treatment for patients that have cancer, not only, again, is there chemotherapy and radiation treatments, but even immunotherapy can be pretty effective, where they can increase the amount of cytotoxic T cells in the body or natural killer cells in the body, because both of them can go after and kill cancer cells. So it can definitely be an effective treatment for some people, for sure. All right, here we have a video that goes through the cell-mediated immune response, talking about helper cells and cytotoxic T cells. So definitely take a look at this, and it's not too long of a video.
White blood cells, called macrophages, lead the body's cell-mediated immune response by engulfing and digesting foreign invaders. Combining peptide fragments from the invaders with some of its own proteins, the macrophage builds a patchwork of antigen complexes on its surface. When smaller white blood cells, called helper T cells, encounter the macrophage, those with matching receptors bind to its surface. These helpers then multiply and secrete chemicals which call different cells into battle, especially the cytotoxic or killer T cells. The macrophage, or an infected cell, activates only those cytotoxic cells with receptors that match the antigen complexes. Stimulated by chemicals from the helper T cells, these killer T cells rapidly divide into an army of clones, all designed to fight the specific invader that triggered the response. When killer cells dock with an infected cell, they bombard it with lethal toxins, then move on in search of other targets.
All right, I hope you enjoyed the video. And again, you can watch it as many times as you wish. It's, I know, extremely exciting. If you like the immune system, immunology, good stuff. So this last slide, before we get into review for today, is an overview of cell-mediated immunity. Again, the focus and emphasis here is on the T cells. So we'll go through this again. Okay, so the red bone marrow, that is where we have the overall origin of T cells, right? This is where they're going to be produced. So this is the origin here when we think of our T cells. Okay, red bone marrow. And then what happens is the T cells are going to go ahead and move to the thymus gland. And this is again where maturation occurs. Two types of T cells that you can have here, produced here, are the CD4 cells is a type of T cell, along with CD8 cells. So those are two types of T cells. So the CD4 cell has a T cell receptor site. And what can happen then, that can eventually travel and can eventually have an interaction with that antigen presenting cell, which displays the peptide fragment of the antigen, which again here we could say it's bacteria, just for an example. That fragment sits within that nice receptor location here of the class 2 MHC protein. The T cell receptor, the CD4 cell, then can dock right here, and based off of that interaction, the CD4 cell becomes activated and realizes that it can go ahead and go after the bacteria. Okay, so they realize that they can go after bacteria, that they're supposed to kill bacteria. And so activation occurs. So then you end up with, again, a large army of clones. So lots of clones are going to be formed. Again, when we think of Star Wars, the Attack of the Clones, lots of clones can be formed. Some of these clones simply become memory cells, but many of them will evolve into what are known as helper T cells, which can travel throughout the body through the blood. We go over here again to the CD8 cell. And again, it has a T cell receptor site, so it can dock onto the fragment of the antigen. We have an antigen presenting cell here, a dendritic cell. Again, that contains the class 1 MHC protein, and the red again would be your antigen fragment. In this case, we can call this a cancer cell, because that's the major target that cytotoxic T cells go after, CD8 cells. So a cancer cell. Based off of this interaction, again, the T cell receptor of the CD8 cell will dock on to the class 1 MHC protein, which contains the fragment piece of the cancer cell. Based off the interaction, you'll have activation, and the T cell, then the CD8 cell, will realize, oh yes, we need to kill cancer, and lots of clones. These are all clones right here. Again, you got a large army of clones which are going to be developed. Some, of course, will simply just become memory cells. But many of them become clones, and ultimately then are going to be called cytotoxic T cells. And again, they can go through your blood and go after and search for cells such as cancer cells to go after and destroy. Again, there's other things they can kill too, but a primary target for the cytotoxic T cells is to kill cancer cells. Okay, so that's our overview here today of cellular immunity. So definitely review that for sure. All right, it's time to review some of the important concepts that we've covered so far in today's lecture. So, number 1 for review, blank immunity contains antibodies that bind to extracellular pathogens like bacteria and inactivate and mark them for destruction. So, what would be the correct answer here? What would be the correct answer here? Okay, and the correct answer for this question here, I would go with choice letter A. Letter A is the correct answer. Humoral immunity. Humoral immunity. Because, of course, this is referring to antibodies and referring to, of course, B cells, right? B cells. Number 2 for review. The origin of the T cells is the blank, while maturation occurs in the blank. So what would be the correct answer for this question here? Okay, and the correct answer here, I would go with choice letter D. So red bone marrow for the origin for the T cells, and maturation, the thymus gland. So, yep. Number 3 for review. Someone who gets the booster shots for the vaccine polio is an example of blank humoral immunity. So what would be the correct answer for this question here? Okay, and the correct answer here, I would go with active, artificially acquired humoral immunity. So that would be choice letter B. So choice letter B would be the correct answer here. Number 4 for review. Blank is an antibody that plays a role in severe allergic attacks and chronic parasitic infections of the GI tract. So what would be the correct answer? Which antibody class are we talking about? All right. And the correct answer, based off these choices, I would go with choice letter C. So the correct answers IgE. IgE. So again, very important to review your antibody classes. Number 5. CD8 cells, when activated, evolve into effector cells called blank, which directly attack and kill other cells, such as cancer cells. So what would be the correct answer here? All right, and the correct answer, I would go with choice letter D. So letter D would be the correct answer here. So cytotoxic T cells would be the right choice, letter D. Well, I hope you enjoyed the immune system. We've definitely covered a lot of ground lately.